Hello friends, welcome to the third lecture of carbohydrate chemistry. This is Harshal Dabne working as assistant professor in Maratha Vidya Prasarak Samaj, Nashik. Friends, in last two lectures we discussed about the introductory matter about the carbohydrates as well their classification into the sugar and non-sugar molecules. So students, in these lectures we are going to focus over the monosaccharides and their reaction. So friends, monosaccharides are nothing but the simple form of sugars which do not undergo hydrolysis. Hence, they also called as the monomer. So these are nothing but the examples of monosaccharides or the monomers. So these four molecules, this one, they are nothing but they contain the six carbon atom. That's why they are called as what say the hexane. Okay, and here this is nothing but said to be the D-glucose. This one is what D-mannose, this one is D-galactose. All these three molecules, they contain the aldehydic group. So this one, this aldehyde, this one is nothing but the another aldehyde, and this one is what aldehyde. So when we do the nomenclature of such type of molecules, so here we found the six carbon atoms present in this molecules, and they have what say the aldehydic group. Hence, it is nothing but the aldohexose molecule. So all these three molecules are nothing but the aldohexose because they contain six carbon atom as well the aldehydic group. So that's why they are included in the family of the aldose. When we move towards what this molecule, so this one is nothing but the D-fructose. Why? Because they contain the ketonic group. So when they contain the ketonic group, so it is nothing but the ketose, and as they contain the six carbon atom, so their name is nothing but the hexoketose. Conversely, when we move towards what this molecule, so it is nothing but D-ribose. So this molecule contain only five carbon atom, first, second, third, fourth, and fifth. They contain only five carbon atoms along with the aldehydic group. So that's why it is nothing but said to be the aldopentose. So these are nothing but the examples of the monosaccharides, or they are said to be the monomers. So what is monomers? They do not undergo the hydrolysis because they are single molecules. They do not further undergo the hydrolysis. That's why they are said to be the monomers. Now we move towards the reactions shown by the glucose. So as we know that in case of the glucose, we have what say the hemiacetal group, that is nothing but the CH2OH, which is easily undergo the oxidation reactions or any condensation reaction. That's why glucose can undergo so many chemicals reaction as well. Glucose, they have what say the aldehydic group, so that's why they undergo reduction reaction also. So the first reactions here we focus, so it is nothing but the glycoside linkage, this formation of the glycoside linkage. So any molecules they having what say the hemiacetal group or the alcohol group is condensed in presence of any other alcohol molecule by means of the strong acid. So we got the glycoside linkage. We further discuss in our next slide. Then next is what say the synthesis of the furano acid. Next one is a sugar phosphate. So sugar phosphate means what? When the phosphate group is reacted with the glucose, so there is a conversion of this molecules into the phosphate, that will be called as the sugar phosphate molecule. The next is nothing but the uranic acids. I already told you. In glucose, we have some groups, means such as what say the aldehydic group, or you can say that the hemiacetal group, which can easily oxidize by means of the oxidizing agent, such as what say the H2O2, and we got what the uranic acid as a product. And finally, the synthesis of the sugar alcohol means suppose we have what say the aldose, means they contain the aldehydic group. So aldehydic group is much reactive and can easily undergo the reduction reaction by means of the NaBH4. And finally we got what's the sugar alcohol. So friends, we try to understand this reaction by considering each example. So first we try to understand the synthesis of the glycoside. So actually glycoside linkage is nothing but said to be the ether linkage shown over here in this diagram. So this is nothing but said to be the ROR group, okay, and that is said to be the glycoside linkage. So this molecule is nothing but the salicin molecules. This is what the glucose molecule. Here we have what's the OH molecule. So when such type of molecule is reacted with what this one, it is nothing but the 2-hydroxybenzyl alcohol. So when such type of molecules means what 2-hydroxybenzyl alcohol is reacted with the glucose in presence of the strong acid such as the concentrated H2SO4, which can act as a dehydrating agent. So what happen when these two molecules are reacted? Which these two? this one and this one, and these two molecules are reacted, okay, in presence of the concentrated H2SO4, so the water molecule can be eliminated and the condensation reaction will be take place, and we got such type of linkage, means what, ROR, that is what the ether linkage, and that linkage is said to be the glycoside linkage. Okay, so this is all about what, say, the glycoside synthesis of the glycoside. This linkage is most common present in the carbohydrate chemistry. Then next is nothing but the synthesis of sugar phosphate. So here we take an example of a D-glucose molecule. This is D-glucose molecules present in the Haworth projection formula, and this is nothing but said to be the hemiacetal group, that is nothing but the CH2OH. So when such type of group is reacted with, say, the subject to the phosphorylation in presence of the hexokinase, which is an we can say that the enzyme. So in presence of this enzyme, so that group is reacted or undergo the phosphorylation at the 6 carbon atom. So this is nothing but said to be the 6 carbon atom, and after that we got what, such type of product. So it is nothing but said to be the D-glucose 6 phosphate. D-glucose 6 phosphate. It is shortly abbreviated as G6P. Is glucose 6 phosphate. This molecules plays an important role in the you can say that the living cell or in this animal, and in this process the 1 ATP will be consumed and converted into the 1 ADP. So see, this is a simple process where, say, this hemiacetal group, means what, CH2OH group present at the 6 carbon atom undergo the phosphorylation in presence of the hexokinase, and in this process 1 ATP will be converted into the 1 ADP. Okay, and at the same time we got this molecule that is called as the D-glucose 6 phosphate, shortly abbreviated as G6P, means what, glucose 6 phosphate. Then we move towards our second our next reaction. It is nothing but the formation of the uranic acid. So friends, we already told about what this hemiacetal group. So this is what hemiacetal groups present over what, the 6 carbon atom of the glucose. So this group is easily oxidized by means of what, any oxidizing reagent. That is nothing but, here we taken the H2O2, which is hydrogen peroxide molecules, acts as the oxidizing reagent. So when we reacted this molecules with hydrogen peroxide, so hydrogen peroxide can oxidize this functional group into this one. So it is nothing but said to be the acid, and similarly we can say that what, after the oxidation of this one, so we got what's the glucouronic acid. So this acid is nothing but said to be the glucouronic acid. So in this way, by using the glucose or by oxidizing the glucose molecules, so we got the uranic acid as a product. Next one is nothing but the formation of sugar alcohol. So obviously we know that in place of the glucose, so glucose they contain what, aldehydic group. This one is nothing but the aldehydic group. Okay, and this one is much reactive. Okay, so when such type of group is reacted with the NaBH4 in presence of the methanol, so this NaBH4 acts as a reducing agent, and that can easily reduces this aldehydic group because it is much reactive, and that's why it will convert it into the hemiacetal, that is what, say, the CH2OH, and that molecule is said to be the sorbitol. So when we do the reduction of any glucose molecule, the aldehydic group will be reduced by means of the NaBH4 in presence of the methanol, and we got the CH2OH, and that molecule is nothing but said to be the sorbitol. So in this way, we synthesize the sugar alcohol by using the glucose molecules, by using simply the any reducing agent, either NaBH4 or any reducing agent. So we got what, the sorbitol, that will be the synthesis of the sugar alcohol. Okay, so students, this is all about what this lecture. In next lecture we are trying to try to explain the reaction and process of disaccharide molecules. Thank you.